everyone. My name is Jack Pistone, and I decided to do my project on aging and longevity. The rise in human lifespan. In only 100 years, humanity has seen a lifespan increase of more than 35%. This has been possible through scientific developments that increase the standard of living. Understanding the role of bacteria in medicine and the ability to utilize computer chips for engineering were large contributors to this increase in lifespan. But what is the next scientific frontier? And it's likely genetics and epigenetics. However, there is an issue with living longer. Despite the immense scientific developments that have occurred, the average human still faces the effects that come with living longer, which is aging. Because with age, the risk of terrible disease increases exponentially. Diseases like diabetes, cancer, neurodegenerative diseases like dementia, loss of bone mass, physical disorders like vision impairment and auditory impairment, and even heart attacks. Additionally, the price of living increases as the healthcare for the individual increases. And that's only for more days spent experiencing the physical pain, limited mobility, and disease of old age. So how can we combat this? It would have to be through slowing the rate of aging. Because we all know that time, aging, and death are inevitable. But is it possible to extend the amount of healthy years an individual can live? Can humans live disease-free lives with the freedom that a healthy body offers? A broader genetic and biological perspective on the aging process could lead to some possible solutions. And in recent years, researchers are discovering that aspects of the aging process can actually be manipulated genetically. Extending the human health span would have larger implications to humanity than extending the human lifespan. And that's simply put that more golden years is better than significantly more bedridden years. So what causes aging? On the right side, we can see some of the factors that are associated with old age in cells. Things like genomic instability, altered intercellular communication, cellular senescence, mitochondrial dysfunction, and telomere attrition, which is the shortening of telomeres. There are numerous biological aging markers seen in older organisms and cells, but these symptoms are the effects of aging. These are not the cause of aging. So this must mean that there is a deeper biological phenomenon taking place, causing these biological symptoms of old age to appear. In the middle of the 20th century, it was theorized before that senescence, biological aging, was the biological effect of the loss of genetic information due to damaged DNA. But now there is a new genetic theory on aging. David Sinclair, he's a professor of genetics and director of the Center for Biology and Aging at Harvard Medical School, believes that aging is the emergent biological symptom of not a loss of genetic information in the genome, but a loss of genetic information in the epigenome. So what does this difference mean? The genome defines the genetic information of the DNA of any cell of an organism. Every single cell has the exact same genetic information. So what is the epigenome? The human body has about 200 different types of cells. And all these cells read the same genetic code, but they somehow differentiate themselves. The epigenome has to do with how this DNA is packaged in the nucleus, and then how the DNA is specifically transcribed or read. So DNA packaging. Simply put, DNA is coiled around proteins that are known as histones. And then the DNA is then marked with methyl groups, and both of these affect the way the genetic code is transcribed. DNA can only be transcribed when it's not wound up, and then methyl groups prevent it from being transcribed. So when DNA is transcribed or damaged, example via radiation, it has to be unraveled from the histones first. And then when this repackaging process occurs, when the DNA is recoiled, it is approximated that 1% of each time it is not recoiled correctly. Histones will attach in the incorrect areas, and then this causes methyl groups to be placed in the incorrect areas. And this actually causes the DNA to then essentially reflect a different genetic code, which then gets read incorrectly and then makes incorrect proteins. So the genetic cause of aging. This incorrect repackaging of the DNA causes a loss of information in the epigenome. A differentiated cell will transcribe the genetic code incorrectly, possibly even becoming a different type of cell, losing its identity. For example, a skin cell could become a brain cell. This is bizarre, but it wouldn't be fatal to the organism. However, if the organism's brain cells begin turning into skin cells, then you have a problem. This phenomenon is likely the main contributor to a genetic cause for aging, and even has been proven. Sinclair and his team can even test an organism for its quote-unquote genetic age, a measure of the methylation, and for instance, the plaque on someone's teeth. This is known as the genetic methylation clock. Here's an image of two mice of the same age from Sinclair's team. The same researchers actually, they genetically engineered a mouse that was 50% older than a mouse of the same age. They did this by, without exposing its actual genome and its DNA to radiation and stuff like that that would cause it to be mutated, the researchers simply severely damaged the mouse's epigenome, causing the loss of information in the epigenome. In this older mouse, it was visibly older, and it was gray, had a hunchback, had mouse dementia, it was blind than the other mouse of the same age. And this mouse didn't just appear physically older, this mouse was physically older. And these mice at the beginning of the experiment were of the same age. So this mouse aged 50% more, simply through its epigenome being altered. This comes to the topic of longevity genes, or as Sinclair's team refers to them as hormetic response genes. So billions of years ago, bacteria developed a very vital biological genetic ability, one where they could live in two modes of existence. When conditions were good, bacteria would be able to grow and proliferate, and then when conditions were bad, they would protect and repair their cells. And these are known as longevity genes. They're parts of the genome that become expressed when an organism is faced with survival adversity. And these genes are still found in our bodies to this day, and they're crucial to the aging process. There's a few different types of longevity genes. The main type are known as sirtuins. The SIR stands for silent information regulator, and these control the information in the cell. But two of the more interesting types are the AMPK and the mTOR. The AMPK is known as the activated protein kinase, and this protein moderates the amount of energy that is taken in to the organism, mostly in the form of sugar. The other type is mTOR, which is the mammalian target of rapamycin. And this protein moderates the amount of amino acids that are consumed. And here on the right, there is just a very complicated protein diagram, protein mechanism. So what does this mean for us? So this study has some pretty broad implications that demonstrate not only the interconnectedness of the body, but something as seemingly unrelated as diet and age. And right now, there are measures that you can take to live longer and healthier through nothing more than making some pretty conscious decisions. The first one would be to avoid DNA damage. You wear sunscreen, avoid radiation. Radiation causes the breakdown of the genome and then the incorrect repacking of the epigenome. And then, of course, avoiding harmful chemicals. Because like we talked about earlier about the methylation of the genome of DNA that prevents it from being transcribed, smoking immediately causes methylation of the genome. And this only accelerates the aging process. The next one is eating less and intermittent fasting. Reducing the intake of protein, amino acids, and sugar put your longevity genes into full force. Intermittent fasting allows for the longevity genes to activate for longer periods of time. So essentially, by having a diet that mimics a diet that would have existed in the wild, the human body puts itself into a stress state that actually repairs its own DNA. So it seems kind of counterintuitive, but the same researchers actually, I'm not sure if it was the same researchers, but there was a study on groups of primates, and they allowed one group of primates to gorge on as much food as they wanted, and then the other group had a caloric restrictive diet. And then they found that the group with the caloric restrictive diet was not only significantly healthier, keener, quicker, and lived longer, but their genetic age was also significantly younger. Next is being uncomfortably hot and uncomfortably cold. And this kind of goes back to the body being in a stress state that would kind of mimic living out in the wild, a time that is not necessarily the best conditions. It puts those longevity genes back into action that then repair your epigenomes. And you could just do this with sleeping with your windows open any time of the year. During the summertime, you can get rid of your air conditioner. It's really terrible for the atmosphere anyways. And you can only use climate control in homes and vehicles when absolutely necessary. Then the last one is high intensity interval training. High intensity exercising puts less persistent stress on your body, in comparison to running. But also high intensity training puts the body into a state of stress where the longevity genes become activated. So the question arises, is this epigenetic damage permanent? Sinclair and his team discovered that these effects are actually reversible through the use of longevity genes. And this big breakthrough was the team's ability to reset a cell's epigenome. So this team not only slowed down the process or the rate of aging, they actually reversed the aging process and put the age of a cell back to an earlier age of that cell. And they did this with they decided to reprogram the retina on the eye of a mouse. They used longevity gene therapy in the eyes of old blind mice. And then they actually reset the epigenome of that mice's retina, and they turned back the biological clock of a 1-year-old mouse to a 2-month-old mouse. And this experiment actually restored the vision to a previously blind mouse, which is pretty incredible. The researchers, what they did is they actually used only 3 of the 4 reprogramming factors to turn back the age of the retina cells in order to prevent the cells turning into basically a large tumor. And on top of that, the researchers were in complete control of the entire experiment. They were actually able to turn on the longevity genes when they wanted to, and then able to turn it off when they needed to. And the team is currently working on resetting an entire mouse's body to an earlier biological age. And then here on the right, you can see the mouse whose vision they cured. So here are some broader implications with the Aurelia aurita, which is the Latin name for the moon jellyfish. So using any gene therapy on every cell in a body is virtually impossible, even with today's technology. However, the answer may lie within the seemingly immortal moon jellyfish. Moon jellies are capable of turning any cell in its body into an earlier version of itself, reproducing asexually, and then creating a genetically identical polyp. So essentially what these jellyfish do is any cell in its body can be reset to an earlier epigenome of itself, and then it can grow, it can break away and then grow into a whole new identical version of that jellyfish. And these jellyfish are known for being immortal. And if humans can gain insight into the underpinnings of this biological process, then we might have the key to true longevity and possibly immortality. So thank you.